Welcome everyone. Today is part 1, RNA Sequencing, Introduction and Kits of the 3-part RNA-Seq webinar series. So let's get started. Let's first begin with an introduction to RNA sequencing. The purpose of this introduction is to understand the workflow for RNA sequencing from RNA isolation all the way to data analysis. We also want to understand key considerations in planning mRNA and total RNA sequencing and consider them before we start processing our samples. We will then take some time to focus on kits specifically, and I do want to remind you that part 2 will be focusing on the best practices, while part 3 of this series will dive into the analysis a bit more. So when we think about RNA, it's often good to think about it in contrast to DNA. DNA is double-stranded, RNA is single-stranded. DNA also shows identity. The amount that is in a cell almost always, it's consistent. It's saying the same thing at the same time. Typically, all cells in an organism will have the same DNA, unless you have a mutation such as in cancer. RNA, on the other hand, it's indicative of activity, right? It is constantly responding to its environment, it's saying different things at different times, and it can vary from one cell type to another, and if there are different types of RNAs. So if we were to label DNA and RNA in respective words, we could, in theory, call DNA stable, while RNA would be considered dynamic. There are a lot of different features of RNA that we can focus on. For example, there is a size that we can look at. We can have microRNAs that are about 20 nucleotides, or mRNAs that can be thousands of nucleotides long. We can also take a look at the shape of the RNA and how it might fold over and form secondary structures. We can take a look at its stability and how long it's present. Lastly, we can see how much is present, looking at individual copy numbers of transcripts within a cell. We can classify these RNAs by function. So the ones that we might be most familiar with are the messenger RNA, ribosomal RNA, and transfer RNAs. These are the ones involved in protein synthesis. We also have RNA species that are more focused on co- or post-transcriptional modifications for DNA replication. And these would be our small nuclear and small nucleolar RNAs. Then we have our regulatory RNAs. These would be microRNA, pRNA, small interfering RNA, maybe long intergenic non-coding RNAs. Together, all of these are considered our transcriptome. Keep in mind that we might have parasitic RNAs as well. So we can use these different functions and modifications of different RNA species to track the specific RNAs that we're looking at. So for example, if we want to look at total RNA sequencing, we might not want to take a look at ribosomal RNA. It's a high percentage of the total amount of RNA, but it doesn't really give us too much useful information. So usually we will want to focus on different areas. So what we'll do in this particular method is we would use a Ribo-Zero or Ribozero Plus kit, which will deplete our ribosomal RNA from our samples. We will then capture the remaining total RNA that's left, and this will be our mRNA and our non-coding RNA. The kits available for this are our TruSeq and Illumina Stranded Total Kit, bundled with either Ribo-Zero or Ribozero Plus. Keep in mind that due to the workflow and our bead cleanup steps, RNAs that are less than 150 nucleotides might be lost during the library prep. So ultimately, we will only have total RNA that's greater than 150 nucleotides, but again, not rRNA. Another common way to enrich for a particular type of RNA We know that messenger RNA and long intergenic non-coding RNAs are polyadenylated. They have that really long, nice A tail at the end, the poly A tail. So we can use beads which have a poly T on them to capture these specific transcripts. If we have viral RNA that are within cells and are polyadenylated, these might be captured as well, depending on the virus type. So the kits that we have available for this particular application are our TruSeq or Illumina Stranded mRNA and our TruSeq RNA V2 kits. If we want to get more specific about the transcripts that we're looking at, we can use sequence-specific probes, which will capture a subset of the total RNA. So for example, our TruSeq RNA Exome kit, formerly known as TruSeq RNA Access, some of you might know it as this name. And this kit has probes specific to the exome that will capture mRNA targets. We know that mitochondrial targets can be captured with this kit as well. Also available is our TruSight RNA Pan-Cancer, TruSight RNA Fusion, and Illumina RNA Prep with Enrichment Tagmentation kits. These kits have probes specific to a subset of mRNA. So if you know a little more about what you are specifically looking for, such as, let's say, cancer genes, for instance, maybe one of these kits would be suitable for your needs. When we're trying to decide what kit to consider, we want to take a step back and say, what are our RNA-Seq applications and what are the questions that we are really asking? We can generally divide them into two buckets, profiling and analysis. Profiling is going to be more counting. So what is the gene expression or changes in gene expression in response to treatments or various cell types? For mRNA, we can take a look at gene expression profiling or allele-specific profiling. If we're looking at mRNA, we can count the different mRNA transcripts within different cells or treatment groups. We can also do this for viral RNA. In terms of analysis, the question is, what is the sequence and are the sequences different? So the mRNA, we can look at whole transcriptome assembly. Maybe we're looking at some novel fusions. We can look at biomarker discovery or novel transcript discovery. So this would be looking at the sequence itself rather than counting how many transcripts are available. We can also look at genome annotation or alternative splicing. We can characterize link RNAs, study small non-coding RNAs, and possibly encounter biodiscovery if there is an RNA-expressing virus. The common RNA-Seq workflow is going to start with RNA isolation, and we are going to discuss this more in just a bit. Then we'll move on to library preparation. Once we've QC'd our libraries, we want to move into sequencing, and we will discuss which instrument is best suited for our specific applications. And then we're going to talk a little bit about the data analysis. But first, let's focus on that RNA isolation, which is really important for RNA sequencing. We are often asked, what kit should I use for my RNA isolation? And while we have no official recommendation, we know that customers have used the MasterPure family of kits by Lucigen. They have also used QIAGEN, Ambien, Trizol, etc. The main point here is that we want good yield and high quality material. We also want to make sure that we are capturing the right species. So if you're looking for microRNA, we want to make sure that the extraction kit that we are using is going to be able to capture microRNAs. So what we want from RNA isolation kits is good yield, high quality material, and as always, check the literature to make sure that we are capturing the right species. Our RNA input quality is going to indicate which kit can be used for our needs. Earlier, I briefly talked about some of these kits and their different applications. If we have high quality input RNA, we are able to use any one of these kits. And what we mean by high quality is that we want a RIN, or also known as RNA integrity number, to be greater than 8. And this applies to mammalian species only. So if you're working with another species, check the literature to see what would be considered a high quality RNA RIN value. Not all kits are compatible with FFPE material. And if we do have FFPE samples, or we have a RIN value that is less than 8, we recommend using either the TruSeq or Illumina Stranded Total RNA, Exome, pan-cancer, or Fusion kits. And these have been developed to be well suited for FFPE samples. Keep in mind that input quality is the #1 most important factor for RNA sequencing, so we definitely want to check that input quality. When dealing with RNA input amount, there are going to be different input requirements for our different kits. So as I mentioned previously, if using TruSeq RNA V2, stranded mRNA, or stranded total RNA, you can use your total RNA. We also have the option of using previously purified RNA. So if you have a method of purifying your RNA that you're comfortable with, you can use purified material for stranded total kits. So how are we going to quantify this material? We recommend using a fluorometric-based method, something like a RiboGreen or Qubit. We find that nanodrop is not the best method for measuring input quantity, as it will likely overestimate, especially if we have not DNase treated our sample efficiently. And as you may have inferred, we recommend a DNase treatment, which is often included in most RNA isolation kits. Keep in mind that DNA could affect accurate input quantification, or could also show up in our library prep downstream as well. So how do we know if our sample is high quality or not? One thing that we recommend is to run an RNA chip on the bioanalyzer. We should be able to see clean ribosomal peaks, and it should also give us an RNA integrity number, that RIN number. With this information, we can determine what our quality actually is. Alternatively, we can run a formaldehyde 1% agarose gel stained with ethidium bromide to see if those RNA bands are intact. Another reason why RNA input quality is so important is, in addition to letting us know what kit is available for us to use, it also informs us of how much input we need. If we have high quality input, we can use 100 nanograms to 1 microgram for our TruSeq mRNA and TruSeq Stranded Total kits, 10 nanograms for our enrichment kits, such as TruSeq RNA Exome, pan-cancer, and RNA Fusion, or 25 to 1,000 nanograms for our Illumina Stranded mRNA prep ligation kit. As you can see, our newly launched Illumina Stranded kits accommodate a lower input amount. If we have low or variable quality, we may need to use a little more input to get even coverage and good yield from our library prep. So in this case, we would still use 100 nanogram to 1 microgram for TruSeq Stranded Total, and as low as 10 nanograms for Illumina Stranded Total RNA. You will notice, however, that TruSeq Stranded mRNA is not recommended for use with low or variable quality input. And if we're looking for enrichment kits, here we'll use a bit more. We'll probably use about 20 to 100 nanograms, depending on the level of degradation and quality of our RNA. We can use this with RNA Exome, pan-cancer, and RNA Fusion kits. We can also use 10 to 1,000 nanograms for our RNA prep enrichment workflow. We do have a great online tool, the Library Prep and Array Kit Selector. The link is provided here, and this tool can assist you in determining which one of these kits is best for your experiment. So once we have our RNA extracted and we know the quantity and quality, we can go ahead with our library preparation. For the RNA purification, usually we'll take one of two approaches. Either we will enrich for the RNA species that we want, or we will deplete the RNA species that we do not want. So for example, a poly-A selection used with TruSeq RNA V2 and Stranded mRNA kits will use the poly-T beads to capture RNA transcripts that are polyadenylated. We will want to make sure that our RIN value is greater than 8, as previously mentioned. If the RIN value is less than 8, this may indicate variability across the sample and can lead to various degrees of 3' bias. That is why we want to have a high RIN value, so material that's really intact. Alternatively, Ribo-Zero or Ribozero Plus depletion uses rRNA probes to deplete the samples of rRNA. Our Stranded total RNA kits use this approach, and these kits are recommended for those RIN values less than 8 or variable RIN values. So any sample with a low RIN value, we've seen RIN values as low as 3. We typically do not see any detectable 3' bias using this depletion approach. This also has the benefit of capturing non-coding RNAs if this is of interest to you. Because we're capturing more RNA species, there is more sample to pull through the prep. So it requires twice as many reads to achieve the same coverage as a poly-A selection approach would. The other option that we have is a post-library enrichment approach used in TruSeq RNA Exome, RNA Fusion, RNA pan-cancer, and the Illumina RNA prep with Enrichment Tagmentation kit. And again, these are recommended for those samples of RIN value 8 or less, or also known as FFPE samples. After we do our RNA selection or Ribo-Zero depletion, we need to convert our RNA to DNA. So what happens is that after the poly-A selection or Ribo-Zero depletion, we will fragment and randomly prime our RNA transcripts. We will then proceed on to first strand synthesis and second strand synthesis. Our method of converting RNA to DNA provides the most quantitative and uniform coverage on the market, as described by the Broad study listed here, which did a comparison of different methods of preparing cDNA for sequencing. Ultimately, it shows that our methods get more uniform coverage across the gene. And this can be important for annotation or alternative splicing. Some key QC points for final libraries. Once we finish the prep itself, we'll want to check it on a bioanalyzer. We want to make sure that the size is the appropriate range that we've targeted. We also want to look for any unexpected peaks. We recommend to quantify using qPCR. The qubit, BA, and nanodrop are not as accurate for RNA sequencing. qPCR is the best method to avoid any unexpected cluster density issues because it's going to quantify only what is going to cluster on the flow cell and nothing extra. If you want to learn more about best practices for RNA library preps, please tune into part 2 of this webinar series, which focuses specifically on best practices. So now that we've QC'd our library, we'll run it on the BA bioanalyzer. And if it looks good, we're going to move on over to sequencing. And one question that we get a lot is coverage. What coverage depth do I need for my RNA-seq analysis? We know that more coverage will give us a greater confidence about the sample sequence. So let's say that the sequence in the gray bar is our reference sequence, and our blue transcripts are going to be our reads that we get from our sequencing experiment. If we had one level of coverage and we see that there is a change from A to C, we wouldn't know if that was an error in alignment, maybe an error in sequencing. We wouldn't be confident that this is a real change in sequence. But if we sequence further and continually see this difference, then we are much more confident that this is indeed supposed to be a C at this specific position for our sample. And you'll notice that in the DNA sequencing world, we talk about coverage in terms of how many X. Is it 30X or 100X? For RNA sequencing, we are using the absolute number of reads. And part of this is because RNA is dynamic. So there will be varying amounts of the transcriptome in various cells. So it's hard to say a coverage for something that is varying, which is why we use the number of reads itself. When we're looking at differential expression, we can gain confidence if we increase our number of total reads. In this particular graph, each dot represents the expression of one gene. The X and Y axis will be 2 replicates. Now, we would expect there to be a solid line across the XY axis. But as we go to the lower region of gene expression, it tends to fan out a little bit. And if we increase our number of reads, some of those low expressors move up to the higher expressing region, and we can then better tell if something is an outlier or if it just has a low number of reads, which is why we're going to be seeing variability. Because we know that genes are present in different copy numbers, it's important to get the right number of reads in order to obtain confidence in what our gene expression actually is. So more reads should result in fewer genes being in the quote-unquote low section here. So how many reads are actually needed? So let's say we are looking at an mRNA, so libraries that have been poly-A selected. If we are looking at RNA profiling or counting the gene expression within the cell, we could use greater than 10 million reads per sample based on a human-sized transcriptome. If we wanted to go a bit deeper and do a transcriptome analysis, we would need a bit more, so say 25 million reads per sample. And these are just general numbers. We highly recommend going to the literature to see what would be best for your species and your application. Keep in mind that these are also specific for mRNA samples. So if we have Ribo-Zero Plus or Ribo-Zero depleted samples, if they are high quality, we'll probably need about 2 times more the number of reads. For Ribo-Zero depleted libraries with low quality or FFPE samples, we will require a bit more, so maybe 4 times as many reads. And part of the reason for this is that FFPE samples tend to preferentially lose their exonic content. So we want to increase our reads, our read depth specifically, to get more confidence for those sequences. For RNA Exome, pan-cancer, and fusion kits, we have a set number of reads that we would recommend. For RNA Exome libraries, 25 million reads per sample. For RNA pan-cancer or RNA fusion, 3 million reads per sample. And again, these are general estimates to be used as a reference. We should always go to OnCode guidelines, which is a great place to start, and definitely check the literature to see what is important for your specific application. In terms of run type, we often recommend a single read for profiling-based applications and pair-end for analysis-based applications because we know that pair-end reads will decrease duplicate percentage, increase alignment percentage, and give us more uniform coverage across genes. Because our TruSeq Stranded mRNA and TruSeq Stranded total kits do give us stranded information, we know that read 1 has better coverage at the 3' end, and read 2 has better coverage at the 5' end. So if we go ahead and perform pair-end sequencing, we will be able to get better coverage at both ends. Pair-end also helps with novel transcript discovery and gene structure characterization. Keep in mind that we will want to ensure that our analysis software can handle pair-end data. While most aligners do well with pair-end data, it's always great to check. And what do we mean by decreasing our duplicate percentage? Let's take a look at this reference genome. The exons are the boxes in gray. The blue boxes are the reads that came off of our sequencing run. If we were to do a single read for all 3 of these transcripts, most likely we would not read through our insert. So if we did a single read 1/75 run, let's say, it might look like we have the same start and stop site for every one of these reads. But if we go ahead and do the pair-end run, we would notice that we have a different stop site for each one of these transcripts. So we will now know that these are not duplicates, and they are in fact coming from unique reads. In terms of read length, our general recommendation is 2 by 75. This is a very general recommendation, and it will vary based on the application. So you can use this as a reference, but always check the literature. So now let's move on to kit options for RNA sequencing. We have 6 mRNA library prep kit options. The differences between these kits lie in how they capture RNA, the output, and the analysis that can be performed downstream. These kits are TruSeq RNA V2, TruSeq Stranded mRNA, Illumina Stranded mRNA Prep Ligation, TruSeq RNA Exome, the Smarter Ultra Low Kit in conjunction with our Nextera XT kit, and the SureCell WTA3' kit. I will not be speaking about the SureCell WTA3' kit today, as it is covered in the single cell sequencing webinar. So if you're interested in that, please check out that webinar. We will start with TruSeq RNA V2. This is considered our original kit and has been widely used for many years. We know that mRNA is polyadenylated, right? So it's polyadenylated. We use the poly-A tail on the 3' end to capture mRNA. To do so, we will utilize beads that have a poly-T tail, which will complementary bind to the A's on the poly-A tail. We will then go into fragmentation and random priming. The reason that we have to fragment our material is that our sequencers, they can't sequence very long pieces of DNA. So we have to bring it to a more reasonable size for the instrument to sequence. We will then enter first and second strand synthesis. At this point, we will have cDNA, which is essentially double-stranded DNA. We will then create blunt ends with end repair, pop an A onto the end during A tailing, and this A will be used to ligate the adapter onto the fragment. Keep in mind that this is a TruSeq chemistry, which means that we will utilize a ligation-based method for adding the adapters. Once the adapters have been added, we will have a sequencing-ready fragment. The information presented in this slide is divided into 3 parts: input, protocol, and output, and I will keep this formatting the same throughout the presentation. So here we can use 2 input types. If we use total RNA, we can use 100 nanogram to 1 microgram. We will want to use high-quality material from eukaryotes. And the reason that we want high-quality material is because we are using that poly-A tail located on the 3' end for pull-down. If we were to use degraded material here, it could be fragmented or shorter in length. Therefore, we could have 3' bias because we would only see the area on the 3' end near the poly-A tail and not the areas in the middle and 5' end. Those would not be pulled down. By having high quality material, we will be able to pull down a longer intact mRNA. The second type of input that can be used is previously isolated mRNA. If using isolated mRNA, you can use 10 to 400 nanograms of high quality input, but you will skip the poly-A selection step. This is a non-directional workflow, which means that if knowing sense versus antisense information is important to you, this may not be the kit to use. You can multiplex up to 24 samples, and it requires 4.5 hours of hands-on time. Our output here is our poly-A selected RNA and mRNA. Now you may say, what if stranded information is important to me? What kit do I use? The TruSeq Stranded mRNA kit may be an option. Now this workflow is very similar to the TruSeq RNA V2, so I'm only going to highlight the areas that are different. So we will start with our poly-A selection, fragment, prime, and go into the first strand cDNA synthesis. Now the difference comes in at the second strand synthesis step. At this step, we will incorporate DUTPs instead of DNTPs. This will have an effect on the PCR amplification step, where the DNA polymerase that is used is non-permissive, meaning that it will not PCR past the U from the DUTPs. This way, we will know that the material which is being amplified is from the top strand. The input and output are the same as RNA V2. What is different is that this workflow is directional due to the incorporation of those DUTPs. Both a 24-sample single index and a 96-sample dual index multiplexing options are available for this kit. Now, what if TruSeq Stranded mRNA sounds great, but you just don't have that much input? Then our newly released Illumina Stranded mRNA prep may be a good consideration. The workflow is very similar to TruSeq Stranded mRNA with two key differences. One, it can accommodate a lower input, and two, it utilizes anchors to add its indexes to the insert. Strandedness, again, is introduced at the cDNA Second Strand Synthesis Step, just as in TruSeq Stranded mRNA, where DUTPs are incorporated into the second strand in place of DTTPs. Since the polymerase cannot incorporate past the first DUTP, the second strand is essentially quenched during this amplification step. Now, the RNA index anchors, these are a new addition to the workflow. This requires an RNA anchor intermediate, which introduces a T-Overhang. And this anchor enables compatibility with at least 384 unique dual indexes. The anchor itself is a Nextera sequence, identical to Tagmentation adapters, plus a T for the ligation compatibility. The impacts of this T-Overhang, which creates a low diversity cycle during sequencing, are very negligible to analysis, as only some impact to run quality may be seen, in which case there are two options that we can use. The first action that we can take is to trim the first cycle, highlighted in the bulletin entitled Trimming T-Overhang Options for the Illumina Stranded mRNA and Illumina Stranded Total RNA Workflows. The second is also a trimming option, but it utilizes a different method with the sample sheet. As previously mentioned, Illumina Stranded mRNA prep ligation accommodates a lower input amount starting at just 25 nanograms of high-quality RNA, which we'll use for poly-A selection. This is a stranded kit which will support a plexity of 384 samples. Now, what if you want stranded information, but you still don't have enough input? For this scenario, you can use the TruSeq RNA Exome kit, formerly known as TruSeq RNA Access. In this workflow, we will create libraries of all RNA present. We will fragment, prime, add adapters, and create full libraries using the TruSeq Stranded mRNA workflow, just skipping the poly-A selection step. We will use sequence-specific probes to bind to sequences for the whole transcriptome, and we can then utilize streptavidin beads to bind to the probes and pull down the libraries of interest. For this workflow, we have the option of using high or low quality input. If using high quality, we can use 10 nanograms. If using low quality, we will have to use a bit more, 10 to 100 nanograms, depending on the DV200. One quick note, when I say DV200, this simply means the percent of fragments that would be greater than 200 base pairs. This kit is compatible with only human samples, as the probe sequences that are used are against the human transcriptome. Since the upstream library prep workflow is done with TruSeq Stranded mRNA, it is stranded. There is the option to multiplex up to 24 single index samples, and our output is the coding transcriptome. For ultra-low input, you can use the Smarter Ultra-Low kit by Takara in conjunction with our Nextera XT kit. In this workflow, you will use the Smarter Ultra-Low kit, again, that's by Takara, to perform whole transcriptome amplification. You will then take 1 nanogram of that cDNA to put into your Nextera XT library prep kit. The Nextera chemistry utilizes an enzyme for fragmentation and adds adapters via PCR. Remember that TruSeq adds adapters via ligation, Nextera adds adapters via PCR. You can use 10 picogram to 10 nanogram of high-quality total RNA with this workflow. 10 picogram is typically the amount of RNA that you can get from one cell. and this kit has been used for single-cell work. You will need 2 kits for this workflow. Again, the Takara Smarter Kit to generate the double-stranded cDNA and the XT kit for the library prep. And because we are using the Nextera XT workflow for library preparation, we can use the dual indexes from this kit to multiplex up to 384 samples. Keep in mind that this workflow is not stranded. And here we have compiled the key features that are different between the kits just mentioned in the matrix. You may use it to look at features that are important for your experiment. For instance, if stranded information is important to you, you may want to consider our stranded mRNA kit. If you have FFPE material, you may consider the TruSeq RNA Exome kit. If you only have 50 picograms of input, you will need to use the Smarter Ultra-Low kit with our Nextera XT workflow. Some other factors to consider: it's always important to consult the literature. Keep in mind project continuation. If possible, it's likely best to continue using the kit that has already been used for a project, so we don't want to switch mid-project. And lastly, take into account cost per sample. Up-to-date information and details can be found on the library prep kit selector and support pages listed here. For more information on the Smarter Kit, I will refer you to Takara's website. Now let's look at whole transcriptome library prep kit options. There are 2 main kits, TruSeq Stranded Total RNA and Illumina Stranded Total RNA Ligation, that we can use for this purpose. The TruSeq Stranded Total RNA workflow is the same as the TruSeq Stranded mRNA workflow. The only difference is that instead of pulling down what we are interested in with poly-T beads, we are pulling out what we don't want. rRNA can be abundant in a sample, and it doesn't really provide much information for sequencing, as I previously mentioned. So we deplete the rRNA before going into sequencing using sequence-specific probes against rRNA. After rRNA depletion, we will skip the poly-A step and enter the workflow, which is the same as previously described. While the protocol and input amount is the same as TruSeq Stranded mRNA, this workflow we can use FFPE material. And the reason why is because we are removing our RNA, but leaving everything else behind. So even if our RNA is degraded, we will still retain it. We will not see the 3' bias as we would with a poly-A selection. This kit supports Ribo-Zero, HMR Gold, Plant, and Globin. And for those of you who are wondering what the difference is between HMR and Gold, Gold includes probes against mitochondrial rRNA as well. If you are not using a sample type listed here, you still have options. You can use a third-party or custom rRNA removal of your choice to deplete the rRNA up front. And once the rRNA is depleted, you can go into the TruSeq Stranded mRNA. And yes, that is mRNA. This is not a typo. Workflow, skipping the poly-A step. This workflow is compatible with human, mouse, rat, Gold, Plant, or you can use your custom rRNA removal system. If using already rRNA depleted RNA, you can use 10 to 100 nanograms. Again, this is FFPE compatible. And quick note, depending on how degraded your sample is, you can modify the fragmentation time. Recommendations on how to modify the fragmentation time are given within the TruSeq Stranded Total RNA User Guide. Here we have the Illumina Stranded Total RNA Prep Ligation with Ribozero Plus. Again, very similar to the TruSeq Stranded Total workflow, just accommodating a lower input and utilizing those anchors that we previously discussed for the addition of indexes. Unlike its predecessor, which utilizes a probe to bind to the target and a magnetic bead to pull the rRNA down, Ribozero Plus utilizes an enzymatic approach to degrade rRNA bound to the target. So the probes will hybridize to the target, enzyme will cleave the rRNA bound to the DNA, and then it'll be digested. So a different mechanism here. The benefit of this is less rRNA carryover, since there is no bead to accidentally pipette into the next step. Not going into detail with this as I've already covered it, I just wanted to remind you that the Illumina Stranded Total Ligation is a stranded prep. And once again, it does utilize the anchors with the same considerations as previously discussed. This kit can support as little as 1 nanogram when using high-quality material, or as little as 10 nanograms when using low-quality RNA, RIN value, let's say 2 through 8 material. Like the TruSeq version, it does provide stranded information and can support 384 samples, but it has an approximate 3-hour hands-on time. And here we have our matrix again for our whole transcriptome kits. And as you may have noticed, the idea behind the kit is that you will rRNA deplete, then you will make your library. Once again, the literature, project continuation, and cost per sample should be considered. Now, if we have questions about specific targets, we can utilize one of our targeted kit options. We have a total of 6 kits, 4 of which are enrichment-based and 2 of which are amplicon-based. The 2 amplicon-based kits I will not get into today as they are covered in other webinars. So you may say, why use an enrichment-based method over an amplicon method? Enrichment workflows tend to be less challenging for larger targets and can be useful in more exploratory-based studies. The enrichment-targeted RNA kits are Illumina RNA Enrichment Tagmentation, TruSight RNA Pan-Cancer, TruSight RNA Fusion, and TruSight Tumor 170. We'll begin with the Illumina RNA prep with enrichment tagmentation workflow. This is an RNA input tagmentation-based enrichment kit. It's essentially the RNA version of our previously named Nextera Flex for Enrichment kit. This kit converts RNA to cDNA, which it can then tagment, so cut. Once the libraries are made, probes are used to enrich for the libraries of interest. Illumina RNA prep with enrichment tagmentation utilizes eBLTL beads. This is the name of the beads, which are different from BLT beads. So those of you using Nextera are probably familiar with BLT beads. These beads are used specifically to tagment double-stranded cDNA and tag them with the adapter sequences. This kit requires 10 nanograms if using high-quality material and a minimum of 20 nanograms if using low-quality material, depending on the DV200. It accommodates 3-plex enrichments with a 384 post-enrichment plexity and has a roughly 2-hour hands-on time. Output will depend on the targets of the panel that you're going to be using. Now, the TruSight Pan-Cancer workflow may look familiar to you as it's the same workflow as the RNA Exome workflow. The only difference here is that instead of having sequence-specific probes against whole transcriptome, here our probe sequences are targeting the cancer transcriptome. The input is the same as RNA Exome, but the output is different as we are now targeting the cancer transcriptome. Same can be said for RNA fusion, so you'll see a pattern here. As you can see, it's the same enrichment workflow, but here we are targeting fusion-specific transcripts. And thus, our output will reflect that. TruSight Tumor 170 is also an enrichment workflow. It targets 170 genes involved in solid tumor formation, and the RNA portion covers some of these. Now, why do I say the RNA portion? Well, this kit option can analyze DNA and RNA simultaneously, allowing us to cover more genes and variants. So both DNA and RNA can be used as input at 40 nanograms. Like the others, the sequences used in the probes are human-specific. The 170 genes targeted by this kit can be found within the manifest, which is located on the kit-specific support page. Alternatively, if you are interested in one specific gene and want to know which one of our kits contain the gene as a target, you can use our Gene Panel Finder tool. With this tool, you can enter the name of the gene you are interested in, and it will display the kits that may be compatible with this gene as a target. To access this tool, you can click on the following link. And here we have, again, our matrix, but for our targeted sequencing kit option. This matrix provides a nice comparison of all the kits discussed within this webinar in one place. Feel free to use it to determine which kit is most compatible for your experiment. Finally, let's talk about some resources that are available to you. First, we have some resources available on the Illumina website. We have an RNA sequencing page that talks about all types of RNA sequencing. We also have the library prep and array kit selector. You can use the kit selector and click around a little bit to see what kit would be best suited for your needs and input type. We have our support pages available for each one of our kits, also listed here. Then we have those base-based apps. Each one will give you more information on how alignments and assemblies are performed. In addition, we will have a link to all publications, including sequencing methods and review. So you can take a look at different methods, different types of RNA experiments, and our library prep methods poster. So the links are on this slide. We have this 3-part RNA-Seq webinar series, Part 1, which you've just attended, Part 2, Best Practices, and coming up, we will also have Part 3, which will be Introduction to Analysis. Now, I didn't talk about it in this webinar, but we do have a small RNA sequencing introduction and best practices webinar. This is recorded, it's on our website, and it focuses specifically on small RNA. So, highly recommend, if you're interested in small RNA, to check this webinar out, and please, check out our site to see if any other webinars are of interest to you. We have some support bulletins. I have highlighted here the RNA-Seq specific bulletins. I have also provided the link to all our support bulletins, which have a lot of really great information. And with that, I will take any questions.